Hi student, I am Sagar Kumar. Here in this video, in the respiration, which topic is most difficult means okay, glycolysis, Krebs cycle, okay, they are having the more names, but the less names are there in the electron transport system, and they used to feel it very difficult, but it is not so difficult. I will make it you very easy because in the NCRT book also he has not written properly. Apijarically he has written. Now you have to read in this way all the things I covered from the electron transport system. What is electron transport system? The are the transported, and what the use in the transporting, they will pump the protons. That is the thing. And oxidative phosphorylation, the electrons, how they will be generated? They will be sent out from the somewhere, that is oxidized. That oxidized electrons will be used for the adding of the ADP, adenosine diphosphate for inorganic phosphate. It will form the ATP. So it is called as oxidative phosphorylation. The energy is coming from by the electron transport. Whenever the electrons are transporting in this way, they used to pump the protons. That is the main thing. So that will be used for the generating of the ATP. That is the heading. Now, why it is so important and where it is to take place? It used to take place in the inner membrane. Mitochondria will have the two membranes. One is outer membrane and second one is the inner membrane. In the inner membranal space, it will be taken. They are present inside of the inner membrane and in the form of 5 complexes. Complex number 1, 2, 3, 4, 5. Out of these 5 complexes, fifth complex will be used for the generating of the ADP to ATP with the help of a ATPase enzyme. Only 4 complexes will involve in the electron transport. This is a neat bit, remember it. Electron transport will be made in the 4 complexes only. Fifth complex use is generating of the ADP to ATP. So, where it is to take place, electron transport system used to present in the inner membrane of the mitochondria, and it is to present in the 5 complexes. Out of the 5 complexes, fifth complex will be used for the generating of the energy, and 4 complexes will be used for the electron transport. Now we will see how the electrons will be coming out, from where it is coming out, and how it is pumping the protons. Now you see, in the complex number 1, it will be used for the 1AD and FAD are the called, they are produced in the matrix of the mitochondria and in the glycolysis also. These produced, what they are producing, NADH plus and FADH2. So NADH plus and FADH2 are called, they are having the energy. So energy rich molecules, stored energy is present. That stored energy will be released in this electron transport system. How it will be done? In the complex number 1, whenever we are seeing NADH plus is to come in the complex number 1, and it will be received by the flavine FMN, flavine mononucleotide. And flavine mononucleotide, what it will do, it is to split the hydrogen into the electron and proton. So it is the oxidation. NADH plus is oxidized, releases the hydrogen, and hydrogen will be splitted by flavine mononucleotide, and electrons, high energetic electrons will be produced, because whenever we are splitting hydrogen, electrons are very high kinetic energy it is to have. That kinetic energy electrons used to pass received by the iron sulfur complex. Whenever the Fmn2, the electrons are passing to the iron sulfur complex, which is present in the complex 1, the kinetic energy will be used, the electron energy will be used for the pumping of the protons from the matrix which is present inside of the inner membrane to the these protons will be taken from here and it will be pumped into the inner membrane space, outer membrane and inner membrane, whatever the place is there, that is called as inner membrane space. So protons are pumped when NAD is oxidized and by the FMN, and the electron energy will be used for the pumping of the NAD electrons, 4 electrons from matrix to the inner membrane space, and electrons will pass Fmn to iron sulfur complex. From there, iron sulfur complex, electrons will be sent into the inner membrane space and it will be received by the ubiquinone. So electrons are receiving, it is a, and it ubiquinone will be sending electrons to the ubiquinone hydrogen, because it will take the 2 electrons are coming, the 2 electrons will take 2 protons from the matrix and it will become ubiquinol. UQH2 means ubiquinol. Ubiquinone means ubiquinol. Remember it. This is the ubiquinol. And ubiquinol will be oxidized. Again, the proton, whatever the hydrogen is there, it will be splitted and it will send 2 electrons to here and protons to the outside inner membrane, that is the outside of the inner membrane, and that means inner membranal space. Now the complex number 3, what it will do, that electrons received by the complex 3, which we used to call them as cytochrome B C1, means cytochrome B and C are present in the complex number 3. So the electrons which came from the complex number 1 through ubiquinone and ubiquinol will be received by the cytochrome B, which is present in the complex number 3, and passes the electron to the iron sulfur complex. Iron sulfur complex will receive the electrons and then the electrons will be sent to the cytochrome C1. So when the electrons are passing from cytochrome to iron sulfur complex to cytochrome C1, their energy will be used for the pumping of the protons from the matrix to the inner membranal space. This is also important, that is not textbook clearly has not mentioned, but diagram he has shown. This only he will ask in the NEET. So at the same time, when the complex 2 is 1 is working, complex 2 also start working. Complex 2, how it will work? Complex 2 used to take the FADH2. FADH2, where it is formed, that is not given in the clearly in the Krebs cycle. In the text also he has not written, but here he has written that is a succinic acid. Succinate means succinic acid. When it is converting to the fumaric acid, at that time it will be succinic acid will be oxidized and electrons will be hydroproton that will send hydrogen to the FAD, flavine dinucleotide. That is the flavine dinucleotide. Here it is a flavine mononucleotide. Remember it. So flavine dinucleotide will receive 2 hydrogens. When succinate is oxidizing to the fumarate, the hydrogen will come and add the FAD, and FAD will become FADH2. These FADH2, whatever the succinate will coming in the fumarate as forming, will be sending out the that whatever the hydrogens. These hydrogens will be again splitted and forms the 2 electrons and 2 protons. This is also important. But the protons are not pumped in the complex 2 because it is a mononucleotide, it is a dinucleotide. Dinucleotide splitting. is energy taking. So the energy which is generated in the splitting of the flavine adenine dinucleotide will be used for that purpose. So they won't pump any protons from the complex 2 to inner membrane space. This is most important for the NEAT. But the electrons, what it will do? It will go into the, it will enter into the inner membrane, in the membrane, inner membrane, inside of the inner membrane, and it will be received by the ubiquinone, and ubiquinone will be received by the ubiquinone hydrogen H2. The same thing, because here ubiquinone H2 will be, again it will be oxidized and giving 2 electrons and 2 protons. The electrons will be received, again it will be sent into the complex number 3, and it will be received by the cytochrome B. Here you should remember one thing, that is the complex number 3 is receiving electron from the complex number 1 and complex number 2 also. In the inner membrane, whatever are the ubiquinone will be converted into the ubiquinol here and here also. So the thing is, both are the same, but the thing is, complex number 2 is not pumping any protons, but complex 1 is pumping 4 protons. That difference, you remember it. And now the electrons which is received by the from the complex number 2 to the cytochrome B, again it will send into the iron sulfur complex to the cytochrome C1. And these electrons, whenever it is coming out from the cytochrome C1, they in the outside of the, this is the outside inner membrane, inner side outside. Inner membrane outside, there will be a cytochrome which will receive the electrons. The electrons, whenever it is coming, something must be there to receive the electron. That is cytochrome C. Cytochrome C acts as mobile electron carrier. Means this cytochrome C receives the electrons from the complex 3, which came from the complex 1 and complex 2, and these finally reach to the cytochrome C, which is the mobile electron carrier. Now the cytochrome C will carry the electron, because it's a carrier, I am telling. So it will carry the electrons and gives the electrons to the complex number 4. And complex number 4, whenever we are seeing here, after reaching the electrons to the complex 4 through the cytochrome C, it will be received by the 2 copper complexes will work in this complex. First copper complex is that the copper A molecule will take the 2 electrons, and electrons will be sent to the cytochromes, that is cytochrome A and cytochrome A3, and finally again it is the copper also is redox, it can do very easily. For that one, copper is used here, means to stabilize the thing, that is not given in the book. Okay, copper A will receive and copper B will send out the electrons via cytochrome A and cytochrome A3. These two are important. Finally, when the electrons are traveling in this way, the electron energy will be used for the pumping of the 2 protons from the matrix to the inner membrane space. So the electron finally reached and came into the matrix side in the complex number 4. Those electrons will be received by the of O2. What is of O2? One atom. Of O2 will receive the 2 electrons, and it will take 2 protons in the matrix, and it will become 2 hydrogen. 2 hydrogen will be, and 1 atom of the oxygen will become the water. This is the oxidation process, because it is taking the hydrogens and electrons also. So it will be oxidized and forms the water. So this water is formed not directly, it is from the metabolism of these things, so it is called, also called as metabolic water. So H2O is formed. Now what we learn up to now, complex 1, complex 3 are pumping the protons into the inner membrane space of the mitochondria. So the inner membrane space is filled with many protons. So the proton concentration will be increased in the inner membranal space, and this compared to the outside of the inner membranal space in matrix side. So they will create a pressure to go out, and here we will apply the chemiosmotic hypothesis. There also you might have read, that is the photo phosphorylation will be taken place in the thylakoid. Oxidative phosphorylation will be taken place in the mitochondria. That is here only. That is the he is telling. So here the total protons, whatever increase the pressure, it will go through the F0 and F1. F0 and F1 are the two particles of the proteins which are present in the ATP synthase. ATP synthase used to consist of the F0 and F1. F1 is to prepare a facilitated diffusion to the protons through the inner membrane. And F1 is another part of the ATP synthase, is to present in the round shape, and whenever the protons are passing through the F0 channel, it will rotate the F1 particle, and the rotation means mechanical energy will be generated. That energy will be used for the combining of the ADP plus phosphate into the ATP, and the protons as usual it will to reach into the matrix. That he has shown detailedly in the other figure. That is, here the one more thing he has told, that is the 1 NADH plus produces 3 ATP and FADH2 produces 2 ATP. That he is explaining now. That is, whenever the inner membranous space, it is inner membranous space is, that ATP synthase enzyme F1 particle is here, F0 and F1 is to present protrude to the inside that matrix side, and 4 protons whenever they are passing through the F0 through F1, it will produce 1 ATP. How? It is a rotates. It is to rotate. That rotating energy will be used for the combining of the ADP plus phosphate, and it will produce the ATP. And the protons will be sent into the matrix. That whatever we are given, he has given explained. Now we will see how the 3 ATP are produced. Now you see NADH is oxidized and producing electron and passing. How many protons it has pumped? That you see, first complex number 1 it's pumped 4 protons, and complex number 3 it pumped 4 protons, and complex number 4 it pumped 4 protons. Total how many? 10 protons are pumped. So but here he telling 4 protons when traveling it will do the 1 ATP. 10 how many? 2.5. 2 to 3, because it will depend upon some situation. So 2.5 can be considered as 3 ATP. Remember this point also. So yeah, NADH plus H is to produce 2 ATP, 3 ATP. Now, whenever we are seeing the FADH2, which is formed by the succinate to the fumarate formation, it won't pump any protons into the inner membrane space. So it will pump only in the complex number 3, that is the 4 protons, and complex number 4, 2 protons. So how many protons it is pumping? 4 plus 2 only by FADH2 oxidation. So 6, whenever 6 by 4, that is 1.5, that also will be considered as the 2 ATP. That is the reason what he is telling. Now once more I will explain. all the thing. That is, whenever the electrons are traveling, the energy will be released, and the electron transport system is traveling, it will release oxidative phosphorylation. That energy which comes out, it will be used for the adding of the phosphorus to the ADP. So it is called as oxidative phosphorylation. Oxidative means energy is coming, and it is used for the phosphorylation, means phosphate will be added to the ADP. Now, this entire electron transport system, oxidative phosphorylation, used to take place in the inner membrane of the mitochondria. Mitochondria consists of two membranes, outer membrane and inner membrane. So, in the inner membrane, this electron transport will be taken place through the five complexes which are present in the inner membrane, which are present inside of the inner membrane. They are the complex number 1, complex number 2, complex number 3, and complex number 4, and complex number 5. Out of these five complexes, complex number 5 will be used for the ATP synthesis. Only the four complexes are used for the ATP, that electron transport. So now we will see how the electrons are generated and how the protons are pumped for the increasing of the electrochemical gradient in the inner membrane to matrix, means outside of the inner membrane space to matrix side. This will be done in the complex number 1 and 2 will be used. They are using the electron energy for the electron for the oxidizing of the hydrogen. How the hydrogen is coming? That only stored energy in the stored energy molecules, energy rich molecules, we can say that is the NADH plus H plus and FADH2. Where they are produced, NADH plus is produced in the glycolysis 2 molecules from the one glucose molecule, and inside the matrix of the, means that is matrix which is present in the mitochondria, there also it will produce in the outside, before entering into the Krebs cycle and after entering into the Krebs cycle also, they will produce one in the mitochondria, they will produce in the matrix outside the Krebs cycle 1 NADH plus, and inside the Krebs cycle 3 NADH plus is to produce, that already we studied, and one FADH2 also produced. And their use is, now it will be electron transport system, whatever the stored energy is present in the NADH plus and FADH plus H2 will be released, and that will be used for the synthesis of the ATP. Now we will see complex number 1, NADH plus will be oxidized and will become NAD in the presence of the NADH dehydrogenase, because it is losing the hydrogen. So what it will do, the hydrogens will be received by the FMN, flavyl mononucleotide, which is present inside of the complex number 1. And what it will do, it will passes the electrons to the, in the iron sulfur complex. FES means iron sulfur complex. Whenever it is passing the electrons to this one, the electrons splitted, means hydrogen is splitted to the electron proton. So it will have high energy. That energy will be used for the pumping of 4 protons from matrix to the inner membrane space. This is the important point. Next, so after reaching the electrons in the complex number 1, iron sulfur complex will send the electrons to the ubiquinone, which is present in the inner membrane. The ubiquinone, again it will become ubiquinol by taking the 2 protons, and it will become ubiquinol hydrogen. That is ubiquinol. That word is given in the text as ubiquinone bracket ubiquinol. This is the ubiquinol, remember it. So ubiquinol, again it will be oxidized, because hydrogen is there, it will be oxidized and split into the electrons and proton. The electrons will send to the complex number 3, and these electrons will be received by the cytochrome B, which is the complex 3 contains cytochrome B and C1. So cytochrome B will receive the electrons from the ubiquinol, and protons will be sent. 2 protons will be sent into this one, and 2 more protons will be taken from the matrix. Totally 4 protons will be pumped. 2 protons which is generated, ubiquinol hydrogen is splitting, 2 protons from the matrix side. And here 4 protons are pumped by the electrons which are passing from the cytochrome to iron sulfur complex iron to cytochrome C1 because of the electron energy. 4 protons are in the same time. At the same time, FADH2 also will be oxidized and releases the energy. How it is? That will be done in the complex number 2. In the complex number 2, whenever we are seeing, if the FADH2 is how it is formed, in the text there he has not given, but he has given only one step. But here he has given clearly, that is succinic acid, when it is converting the fumaric acid name he has not given that in the Krebs cycle. But I given in the previous video. That details everything I given in the previous video. That is the aerobic respiration. So succinic acid, when it is converting the fumarate, succinic acid will be oxidized and it will be convert the FAD into the FADH2. The FADH2 loses splits the hydrogen which is present, 2 hydrogens, into the proton and electrons. But the energy is not used. Electron energy which is generated in the complex 2 is not used for the pumping of the proton. This is the most important point in this. And only electrons will be passed to the iron sulfur complex. Iron sulfur complex as usual there. Here also ubiquinol. And again here the splitting of the hydrogen, and it will send the electrons to cytochrome B and passes like this and pumps up 4 protons. So that FADH2 used to pump, not pumping here, but it is pumping in the complex 3. So in this way, what you should understand is the electrons are reaching to the complex 3 from the complex 1 and from the complex 2. But when it is from the complex 1, it is coming, it used to pump 4 protons into the inner membrane space. But here, whenever in the complex 2, it is not pumping any protons, but it is sending the electrons to the ubiquinol, and it is traveling as usual. And finally, the electrons from cytochrome C1 to it will reach to the, it will carry that cytochrome C, which is present outside of the inner membrane. This is also most important point. It is also called as electron mobile carrier. This electron mobile carrier, the cytochrome C, is to carry the electron by receiving the electrons from the cytochrome C. It is to carry and brings to the complex number 4. Complex number 4, here the electrons will be received by the oxygen and it will be converted into the water. So here the electrons which came by the mobile carrier cytochrome C will be received by the copper A molecule, which is the 2 copper molecules are present in the complex number 4 as copper A and copper B. Cytochrome C is to give the electrons to the copper A, and from there to cytochrome A, cytochrome A3, then copper B. It like that it is to travel the electrons which came from here. And whenever they are traveling like this, they also used to pump 2 protons from the matrix to the inner membrane space. So finally the electrons reach to the matrix side and it will be received by the off O2. Off O2 means one molecule, one atom of the oxygen. That oxygen also will take the 2 protons. It will be oxidized, and the electrons it will take and it will form the water. So this is, we used to say, but generated water. So metabolic water is formed. So oxygen, where it is used in the tail end of the electron transport. So it is receiving the electrons which are travel from the entire chain. That is electron transport system. So here and next, what it has done, electron whenever it is transporting like this, it has pumped some protons to the inner membrane space. So the proton, here the thing is chemiosmotic hypothesis will be applied here. The proton gradient, whenever it is formed in the thylakoid from the lumen to the outside stroma, it is to pass. Here proton gradient is formed inner membrane space, and that is electrochemical gradient. We used to say it will be more in the inner membrane space than the matrix. So they have to forcefully they used to move by the diffusion. How they will diffuse? For the diffusion purpose, ATP synthase enzyme will be used. ATP synthase enzyme is a protein. It will present in the two parts. One is the F0 and one is the F1. The F0 particle act as the the protons are used to travel, no? That is called as facilitated diffusion. Channel will be prepared by the F0 particle, which is a part of ATP synthase. Now F0 particle 2, it is forming a channel and it will come out and it will pass through the on the surface of the F1 particle, a part of ATP synthase, and it is in the round shape. So whenever it is passing like this, it will rotate. The headpiece like thing is there, no? F1 particle, it is to rotate. Whenever it is rotating, it will give the some energy, mechanical energy. That energy will be used for the combining of ADP and phosphate will be combined and it will be pressed and it will form the ATP. In this way, ATP will be formed. And during this complete explanation, in the text he has given one more point. NADH plus will be produced 3 ATP, FADH plus to produce 2 ATP. How it is that he explained here. The inner membrane space, whenever they are traveling, inner membrane space, inner membrane, this is inner membrane space, proton gradient is more, and out in the matrix side it is less. So they have to travel F0 particle and F1 particle. He has shown clearly here. And 4 protons, if you are passing through the F0 channel and reaches the F1 particle and goes to the matrix, what it will do? It will rotate the headpiece and ADP will phosphate will be pressed and forms the ATP. Now 4 are sending and NADH plus is forming 3 ATP. How it is? That also you will get that. Now I will explain that. Whenever the electrons are passing through the electron complex number 1, NADH plus, it is oxidized and it will electrons will pump the 4 protons from the complex number 1, and complex number 3, 4 protons, complex number 4, 2 protons. How many came from NADH plus? 10 protons came. 4 plus 4 plus 2. So 10. So 4, 10 by 4. That is a 2.5. We can consider it as 3. Next, FADH2 is to produce 2 ATP. How? FADH2 is not pumping any protons into the inner membrane space in the complex number 2, but it is pumping 4 protons in the complex number 3. So 4 plus here complex number 4, 2, it will become 6. 6 by 4 is 1.5, that is the 2 ATP. This only the electron transport system. Please listen this one once or twice and see this one, and then you read the textbook. You won't get any doubt. You are able to do any type of the answer, whatever he gives in the NEET. More than this much explanation, you won't get anywhere, I am assuring you. So if you like this video, please like the like mark you give, and this type of the explanation you can come to know how I given. I am sure I am given very good explanation, I think, because I know I am having 32 years experience. You subscribe my channel because you are Before this one, I given aerobic respiration. Before that one, glycolysis, and before that introduction part, and before that one photosynthesis. All the five videos I done. All the things, my explanation will be like this. My explanation will be used for the from the base level, means 11th class students. Also it will be used, and NEET we are going the right, that 2026 NEET also. This explanation is very good, no doubt about it. And if you feel like that, share to your friends also, subscribe the channel. And next tomorrow I will release one more video that contains of balance sheet, that is also important one, and amphibolic pathway and RQ, respiratory coefficient. A small video will come. Later on, I will continue with the plant growth and development. With that, entire physiology will be over by the within 1 week or 10 days. Later on, I will start principles of the inherency in the second year, followed by the molecular basis of inherency, followed by the biotechnology. These topics, whatever I completed, they are the hard topics that you know, and I also personally know. These topics, why I chose, because this type of topics was to be covered. The student will get the confidence, and he will read all other things. Whatever I told, primary physiology and other things, 20 bits will come out of 45. So how much useful, and they are the hard topics. These only will decide your seat. So subscribe my channel. Whenever my video is coming, please see that one. Thank you.